Somewhere beneath the Antarctic ice, the ocean holds a temperature just below freezing. The water is dark, still, and almost unchanged for millions of years. On the seafloor, something is moving. It does not move quickly. It does not need to. The environment around it does not change enough to demand urgency. What moves here has adapted to stability, and to pressure, and to cold. What the light eventually reveals is not large by land standards, but it should not be this large at all. It is a spider. Its body is small, but its legs extend outward in long, thin segments, reaching a span wider than a dinner plate. It has no lungs, no gills, no visible respiratory system of any kind. It breathes through its skin. In most oceans, that would be a hard limit. A body built this way cannot grow beyond a certain size. The physics does not allow it. Here, that limit changes. The same organism that would remain the size of a fingernail elsewhere grows far beyond it, not slightly larger, orders of magnitude larger, and it does not happen once. This pattern repeats. Sea spiders are only one example. Across the Southern Ocean, multiple unrelated animals follow the same direction. Larger bodies, extended limbs, systems operating at a scale that should not be sustainable. That is where the mismatch appears, because this environment is not energetic. It is cold, stable, and slow. Temperatures remain just below freezing, with almost no variation over millions of years. Nothing about that should favor size, but it does. These animals are not recent arrivals. They have been here for tens of millions of years, developing in isolation inside a system that has barely changed. Their bodies reflect that. Minimal central structure, organs distributed into the limbs, designs that would not function in most environments. But here they do, because if this were a single case, it could be explained as an anomaly. It is not. It is a pattern, a repeatable outcome produced by a specific set of conditions. In this journey, we look at how Antarctica reshapes the limits of animal size, why sea spiders are the most extreme example of that process, and what this environment reveals about how far biological systems can be pushed under the right constraints. Long before the ice became permanent, beside the silence settled into something unbroken, the southern edge of the world still belonged to a wider system, a place connected by slow drifting currents and shallow marine corridors that allowed life to move, mix, and disperse. That continuity ended with a shift so gradual it would have been invisible within a single lifetime, yet so decisive that it redrew the biological map of the planet. Around 34 million years ago, a final separation occurred between Antarctica and South America, opening a passage of water that had once been closed. The break was not violent, but it was absolute, and once it formed, the consequences began to accumulate without interruption. The new channel allowed a current to form, one that did not wander or dissipate, but instead circled the continent endlessly, driven by the rotation of the Earth and the absence of land to interrupt its path. This became the Antarctic Circumpolar Current, a continuous ring of moving water carrying approximately 130 million cubic meters each second. It did not merely flow, it enclosed, creating a boundary that separated the Southern Ocean from all others. Warm water from lower latitudes could no longer reach the continent in any meaningful way. Heat was kept out, cold was kept in, and the isolation deepened with every passing cycle of the current. Within a relatively short geological span, measured in a few million years, temperatures fell sharply. Ice began to accumulate, first in patches, then in sheets, until the continent became locked beneath a vast and persistent cover. The surrounding seas cooled alongside it, settling into a narrow thermal range that would remain almost unchanged for millions of years to come. Shallow marine pathways that once connected Antarctic ecosystems to the rest of the world were sealed beneath ice and cold, removing any remaining routes for exchange. The organisms already present in those waters did not migrate away. They remained where they were, enclosed within a system that no longer communicated with the rest of the planet. Time continued, but the conditions did not. For approximately 34 million years, life along the Antarctic continental shelf developed in this state of near-perfect isolation. Evolution proceeded without the usual interruptions, without large-scale invasions of new species, without the constant reshuffling that defines most marine environments. Lineages persisted, diverged, and specialized in response to a setting that remained stable to an unusual degree. The result is visible in the present day, where roughly 35% of Antarctic marine invertebrate species exist nowhere else, a level of endemism comparable to island systems, yet extending across an entire oceanic region. The water itself became a defining feature of this system, maintaining temperatures between minus 1.8 degrees Celsius and plus 1 degree Celsius across immense stretches of the continental shelf. This range, narrow and consistent, persisted across timescales that would normally encompass dramatic fluctuations elsewhere. Seasonal changes occurred, but the baseline remained anchored within limits that barely shifted. In most oceans, variability is a constant force, shaping organisms through cycles of change. Here, the absence of variation became the dominant influence, a quiet but persistent condition that shaped every aspect of life beneath the surface. What emerged from this long stability is not immediately obvious from a distance. It does not announce itself through sudden extremes or visible volatility. Instead, it operates through accumulation, through the slow reinforcement of biological traits that are never forced to reset or adapt to abrupt change. Every generation builds upon the last under nearly identical conditions, allowing patterns to extend further than they would elsewhere, unbroken and uninterrupted. Stability, in this context, is not a passive backdrop. It is an active force, shaping outcomes through its refusal to shift. There is a kind of creature whose shape seems unfinished, as though the usual instructions for building a body were set aside halfway through and never returned to. It stands on long, narrow limbs that carry almost everything it needs, while the center remains reduced to little more than a junction, a quiet meeting point for structures that do not belong there by ordinary design. This is the animal often called a sea spider, though the name suggests more familiarity than the organism allows. The resemblance to true spiders is only surface deep, a coincidence of legs and posture rather than ancestry. These animals belong to a group known as Pycnogonida, a lineage so old and so structurally distinct that its place among other arthropods remained uncertain for generations of researchers. Their form resists easy classification, not because it is complex, but because it is almost stripped down to essentials, leaving behind a body plan that feels both ancient and unresolved. At the center sits a trunk so small it cannot contain the full machinery of a typical animal. From it extends a head-like structure bearing a rigid tube-shaped mouth, a proboscis designed for precise feeding rather than flexibility. Surrounding this core are the legs, usually 8, though some species carry 10 or even 12, each one slender, jointed, and disproportionately important. What the trunk cannot hold, the legs must carry, and so evolution has arranged the internal systems in a way that seems almost counterintuitive. The digestive tract does not remain confined to the center. It branches outward, extending into each leg, sending nutrients along narrow channels that run through the limbs themselves. Reproductive structures follow the same pattern, spreading into the legs rather than remaining centralized. In males, a specialized pair of appendages known as oviges extends forward, delicate yet purposeful, serving a single role that defines them. These structures cradle fertilized eggs, holding them in place as development unfolds, turning the limbs into a moving support system for the next generation. Circulation within this body is equally minimal. There is no heart in the familiar sense, no chambered organ driving blood through vessels with rhythmic force. Instead, a simple pumping motion moves fluid slowly through the internal spaces, sufficient for a creature whose needs are modest and whose pace is unhurried. More striking still is the absence of any dedicated respiratory organ. There are no gills to extract oxygen, no lungs to draw it in, no specialized structures devoted to breathing. Oxygen enters passively, diffusing across the surface of the body through the cuticle, a thin outer layer that separates the internal from the surrounding water. This process depends entirely on the gradient between the oxygen dissolved in the environment and the oxygen within the tissues. It is a quiet exchange, continuous and unforced, but also inherently limited. As body size increases, the demand for oxygen rises with volume, while the surface available for diffusion grows more slowly. At a certain point, the balance fails, and the animal can no longer sustain itself. In most oceans, this constraint holds firmly. Sea spiders elsewhere remain small, often no larger than a fingernail, their bodies kept within dimensions that diffusion can support. The architecture of their form does not change, but its scale is tightly bound by the properties of the water around them. Growth beyond that limit becomes unsustainable, not because the design is flawed, but because the environment does not permit it. In the waters surrounding Antarctica, this constraint loosens in a way that is both subtle and profound. The same body plan persists, unchanged in structure, yet the scale shifts far beyond what would be possible elsewhere. The reason does not lie within the animal itself, but within the medium it inhabits, a property of the water that alters the balance between surface and volume, between supply and demand, allowing a form that should remain small to extend outward into something far larger. The explanation begins not with biology, but with the behavior of gases in cold water, a principle that operates quietly beneath every living surface. It begins with a simple rule that belongs to water itself, a quiet principle that operates whether anything is alive to depend on it or not. When a liquid becomes colder, it can hold more gas within it, not by effort or design, but by the basic arrangement of its molecules. As temperature falls, movement slows, space opens, and gases remain dissolved more easily. This relationship, often described through Henry's law, does not change from place to place. It is constant, steady, and indifferent to what it makes possible. In the waters that circle Antarctica, this rule unfolds at the edge of freezing, where temperatures rest near -1.8 degrees Celsius. At that threshold, seawater carries a noticeably higher load of dissolved oxygen than warmer oceans at similar depths, often between 20 and 40% more. Nothing in the biology of the region creates this abundance. It is not produced by the animals that rely on it. It is simply present, maintained continuously by the cold itself, renewed with each cycle of mixing and circulation that passes through the Southern Ocean. For organisms that exchange gases directly across their surface, this difference is not subtle. It reshapes the boundaries of what their bodies can sustain. Oxygen moving into tissue by diffusion depends on the gradient between the surrounding water and the cells within. When that gradient is steep, the flow remains steady. When it narrows, the supply weakens. In most environments, this sets a strict limit on size. As an animal grows, its volume increases faster than its surface area, and the demand for oxygen begins to outpace the ability to absorb it. Beyond a certain point, growth becomes a burden, and the body itself imposes a ceiling. In colder water, where oxygen is more abundant, that balance shifts. The surface remains the same, but the supply is richer, allowing diffusion to keep pace with the needs of a larger body. The constraint does not disappear, but it recedes, extending the range within which growth remains viable. A form that would stall in warmer seas can continue to expand, supported not by a change in its design, but by the properties of the medium surrounding it. This relationship has been studied across many species within the Southern Ocean, revealing a pattern that extends beyond any single lineage. In the early years of this century, a detailed analysis brought together measurements of body size and oxygen availability across a wide range of invertebrates, including the long-limbed forms already described. The results showed a consistent link between the amount of dissolved oxygen in the water and the maximum size these animals achieved. The pattern held across different depths, across distinct groups, and across variations in body structure, suggesting a common environmental influence rather than a shared genetic trait. The findings did not suggest intention or direction. They described a boundary, a limit defined by physics that organisms could approach but not exceed. Within that boundary, size could increase as long as the supply of oxygen remained sufficient to meet the demands of living tissue. The cold ocean, by holding more oxygen, raised that boundary, allowing bodies to extend further than they could elsewhere. Yet this explanation, precise as it is, remains incomplete. It accounts for what is possible, but not for what is chosen. It defines the conditions under which growth can occur, but not the forces that make it happen. An expanded ceiling does not compel an animal to reach it. It only permits the attempt. Within this quiet abundance, something continues to press outward, generation after generation, guiding form toward the limits set by the water itself. Growth, in most environments, carries a quiet penalty. Every increase in size demands more energy, more oxygen, more time exposed to risk, and unless something offsets those costs, natural selection settles toward balance rather than excess. Large forms do not appear without reason. They persist only when the advantages of being bigger accumulate steadily enough to outweigh the burdens that come with it, generation after generation, until size itself becomes a reliable path rather than an exception. Along the Antarctic seafloor, several pressures align in a way that gently but persistently favors expansion. The first of these is the nature of food. Much of what sustains these animals does not drift or swim, but remains fixed in place, growing as dense, layered colonies across the substrate. Hydroids extend in branching forms, bryozoans assemble into intricate lattices, and soft corals spread slowly outward, each anchored to a single point. For a predator moving among them, feeding becomes a matter of contact and coverage rather than pursuit. A longer reach allows access to more surface at once, reducing the need to relocate and increasing the amount gathered within a single position. With each extension of limb length, the area that can be explored expands, and the boundary between effort and reward shifts slightly outward. This advantage accumulates in a setting where movement is slow and energy is conserved. A larger individual can remain within a productive patch longer, drawing from it more completely before moving on. Over time, this translates into a steady gain, not dramatic in any single moment, but consistent enough to influence which bodies leave more descendants behind. The shape does not change, but its scale begins to stretch, guided by the quiet arithmetic of access. A second influence emerges through reproduction, carried directly along the same limbs that define the animal's form. In many species, males take on the role of transporting fertilized eggs, holding them securely for extended periods as development unfolds. This responsibility is not brief. It can extend across long spans of time in cold water, where growth proceeds slowly and patience becomes part of survival. A larger frame provides more space for this task, allowing a greater number of eggs to be carried at once. Each additional cluster represents a future that might continue the lineage, and so even small increases in capacity become meaningful over generations. The connection between size and reproductive output is subtle but persistent. A slightly larger individual may carry a slightly larger clutch, and those offspring, inheriting similar proportions, may repeat the pattern. Over long periods, the effect compounds, turning incremental differences into a directional trend. Size becomes not only a matter of feeding, but a direct extension of reproductive potential, woven into the same structures that sustain the animal in other ways. A third pressure lies in what is absent. In many marine environments, growing larger brings greater visibility to predators capable of breaking shells or overpowering soft bodies. This risk acts as a counterbalance, discouraging unchecked expansion. Along the Antarctic shelf, that constraint has historically been reduced. The range of active shell-crushing predators has been limited, and the interactions that would normally impose a ceiling on size have been less frequent. In such conditions, the usual cost of becoming larger diminishes, allowing other advantages to dominate without the same level of opposition. These influences do not act separately, they overlap, reinforcing one another in quiet alignment. Greater reach supports feeding, which supports growth. Increased size supports reproduction, which supports persistence. Reduced predation lowers the cost of both. None of these forces is abrupt or dramatic on its own, yet together they establish a consistent direction, a gradual movement toward larger forms that continues as long as the conditions remain unchanged. The surrounding water provides the capacity, holding enough oxygen to sustain the expanding body. The environment provides the incentive, rewarding each increment of growth with small but reliable gains. Across long spans of time, these factors combine, shaping an outcome that appears striking when seen all at once, yet emerges from processes that are steady and continuous. And within this same setting, other lineages follow similar paths, responding in their own ways to the same quiet pressures. Seen across the wider seafloor, the pattern does not remain confined to a single lineage or body plan. It appears again and again, expressed through forms that share no close ancestry, each arriving at a similar scale by entirely separate paths. What seemed at first like an isolated outcome becomes, with distance, a consistent theme woven through the living fabric of the Antarctic shelf. Among the crustaceans, certain isopods move slowly across the sediment, their bodies segmented and armored in a way that recalls familiar shapes from warmer coasts, yet expanded far beyond those proportions. Where related species elsewhere remain small, often no more than a few centimeters in length, these individuals extend to sizes that approach 20 centimeters, their presence altering the sense of scale along the bottom. The proportions remain recognizable, but the dimensions shift, as though the environment has allowed each segment to grow without interruption. Elsewhere, ribbon worms stretch along the substrate, their elongated forms reaching lengths that would be improbable in other oceans. Some extend beyond 2 meters, slender and continuous, their movement subtle and unhurried. In more temperate waters, similar organisms tend to remain closer to a fraction of that length, constrained by conditions that limit how far such bodies can extend while remaining functional. Here, those constraints appear softened, allowing the same design to unfold further. The seafloor itself is anchored in places by organisms that grow in silence over spans of time that extend beyond human reference. Sponges rise from the substrate in massive structures, some reaching the size of small vehicles, their surfaces textured by the slow accumulation of living tissue. Growth rates are measured in increments so slight they become meaningful only when considered across thousands of years. Certain individuals are estimated to have persisted for more than 10,000 years, their forms shaped by a continuity of conditions that rarely shift. Other familiar groups follow similar trajectories. Sea urchins expand beyond the sizes typically seen elsewhere, their tests broader, their spines longer, their movements steady. Starfish extend their arms across wider spans, covering more ground with each slow motion. Amphipods, often overlooked in other environments due to their small size, appear here in larger forms, their bodies scaled upward while retaining the same basic structure. Each group arrives independently at a similar outcome, shaped by pressures that act consistently across the community. Even among the drifting populations, the pattern holds. Antarctic krill, known scientifically as Euphausia superba, reach lengths of around 6 centimeters and live for several years, far exceeding the lifespan and size of their relatives in warmer seas. Their swarms move through the water column as a living system, sustained by conditions that support both growth and longevity in ways that are less common elsewhere. What connects these examples is not shared ancestry, but shared environment. These organisms belong to different branches of life, separated by evolutionary distances that stretch back over 500 million years. Their last common ancestors lived in oceans that no longer exist, yet in this one region, they respond in parallel, each adjusting its scale in response to the same underlying conditions. The repetition of this pattern across such diverse forms suggests a cause that lies outside any single lineage. The influence comes from the water itself, from the consistent presence of oxygen in a state that supports extended growth, from temperatures that remain within narrow bounds, and from a stability that allows incremental changes to accumulate without interruption. The result is not an isolated anomaly, but a coordinated expansion across an entire ecosystem, expressed differently in each organism, yet driven by the same quiet factors. This environment does not resemble the kinds of extremes often associated with life at its limits. It does not rely on bursts of heat or chemical intensity. Instead, it offers a steady abundance, a set of conditions that support life in a way that is both unusual and consistent. The seafloor beneath the Antarctic waters becomes a place where growth is not constrained in the usual ways, allowing forms to extend into dimensions that elsewhere remain out of reach. To understand how this unfolds in detail, it becomes necessary to consider the structure of the seafloor itself and the communities that occupy it, which differ from common expectations in ways that become clearer with closer observation. From a distance, the seafloor here is often imagined as empty, a quiet expanse of rock and shadow where little has taken hold. That image persists because so few have seen what lies beneath in detail. When cameras descend into these waters, the impression shifts almost immediately, not through sudden spectacle, but through the steady accumulation of life across every surface within view. The Antarctic continental shelf supports a density of invertebrate life that rivals, and in some places exceeds, many of the most biologically active marine environments elsewhere on the planet. This richness is not distributed in isolated patches, but extends across broad stretches of seabed, forming communities that appear continuous, layered, and deeply interconnected. The cold, stable water that surrounds the continent allows growth to proceed slowly and without interruption, and over long periods, this continuity produces structures that fill space in ways that defy expectation. Hydroids form one of the most visible components of this landscape. These colonial organisms, related to jellyfish, grow upward from the substrate in branching forms that resemble delicate underwater shrubs. Some reach heights approaching a meter, their fine structures extending into the water column and creating a vertical dimension to the seafloor that is not immediately apparent from above. Within these branching networks, smaller organisms find shelter, surfaces for attachment, and opportunities for feeding, turning each colony into a framework that supports additional layers of life. Interwoven among them are bryozoan colonies, building rigid structures from calcium carbonate that accumulate over time into complex formations. These colonies expand outward, linking with neighboring growths to create reef-like patterns that spread across the substrate. The architecture is intricate, composed of countless small units working together, each contributing to a larger structure that alters the physical character of the seafloor. These formations provide both habitat and feeding grounds, reinforcing the density of life that defines the region. At greater depths, soft corals extend into this same space, forming gardens that stretch across hundreds of meters. In areas such as the Weddell Sea and around the South Sandwich Islands, these communities display a level of structural complexity comparable to well-known tropical reefs, though shaped by entirely different conditions. Their forms are adapted to low light and steady currents, growing in patterns that maximize exposure to passing water while maintaining stability over long spans of time. Sponges add another layer to this environment, their growth measured not in rapid expansion, but in persistence. Over thousands of years, they accumulate into fields that cover large areas of the shelf, forming dense, textured surfaces that appear almost continuous when viewed through remotely operated cameras. Some of these individuals have persisted for more than 10,000 years, their structures recording the passage of time through incremental growth that rarely pauses. Together, these communities create a foundation that sustains the larger organisms moving among them. The same conditions that support their development, the consistent availability of oxygen, the absence of rapid change, and the long, uninterrupted timescales also support the expansion of the animals that feed upon them. The relationship is direct and continuous, with each layer of life contributing to the stability of the system as a whole. Observations from recent expeditions have reinforced this view. During a survey conducted in 2022 by the Schmidt Ocean Institute, remotely operated vehicles captured footage of seafloor communities around the South Sandwich Islands that appeared unusually dense and diverse, even to researchers accustomed to polar environments. Within a limited series of dives, 3 previously unrecorded species of sea spider were documented, found in areas that had received little prior attention. Such findings suggest that what has been observed so far represents only a portion of the full system. The extent of the Antarctic shelf is vast, and the areas that have been directly examined remain comparatively small. Each new survey adds detail, but also reveals how much remains beyond direct observation. Understanding the scale of what is still unseen requires looking beyond individual discoveries to the broader scope of exploration itself. Measured against the scale of the continent itself, the region beneath the surrounding waters extends outward in a quiet expanse that is difficult to fully hold in mind. The Antarctic continental shelf alone covers approximately 4.6 million square kilometers, an area that surpasses the landmass of Australia, yet much of it remains beyond direct observation. It lies below a surface that is often inaccessible, beneath conditions that limit both time and reach, leaving vast sections known only through inference rather than sight. The tools used to explore this space have advanced steadily, yet their presence within these waters remains limited. The total number of hours spent by remotely operated vehicles below depths of 200 meters across Antarctic regions, even into recent years, can still be counted in the low hundreds. Each descent captures only a narrow corridor of terrain, a brief passage through a much larger landscape that continues in all directions beyond the frame. The images gathered provide detail, but they do not yet form a complete picture. Efforts to map the seafloor have accelerated in recent decades, with more coverage achieved in the last 20 years than in the preceding 150 years of Antarctic research combined. Despite this progress, the portion that has been charted remains only a fraction of the whole. Large areas persist as outlines and approximations, their contours suggested by indirect measurements rather than confirmed by direct visual record. The scale of what remains unobserved continues to exceed what has been documented. Beyond the continental shelf, the depth increases into regions that are even less explored. The South Sandwich Trench reaches down to approximately 8,428 meters, forming one of the deepest points within the Southern Ocean. Visits to these depths have been rare, limited to a small number of expeditions that have briefly touched this environment before withdrawing again. Each visit adds to the record, yet leaves the broader expanse largely undisturbed. The earliest systematic attempts to study Antarctic marine life began in the late 19th century during the Challenger expedition, which marked the first comprehensive oceanographic survey. The organisms brought to the surface during those efforts were unfamiliar in both form and scale, challenging expectations shaped by more accessible seas. Even then, the significance of what had been collected was not fully recognized, in part because the specimens often arrived damaged, and in part because comparable data from other regions was limited. Through much of the following century, certain organisms remained in the background of Antarctic research. They were recorded, preserved, and cataloged, yet rarely examined in detail. The broader implications of their size and structure were not immediately apparent, and without a framework for comparison, they remained isolated observations rather than components of a larger pattern. It is only within the past 30 years that these fragments have been brought together into a more coherent understanding, revealing connections that were previously unnoticed. Researchers focusing on these animals have continued to expand the known diversity within this environment. Among them, Claudia Arango has contributed significantly to the identification and description of Antarctic sea spider species, adding numerous entries to a catalog that is still incomplete. The number of distinct forms already documented suggests that many more remain unrecorded, existing within regions that have yet to be examined closely. The possibility remains that individuals of considerable size have not yet been observed, moving across sections of the seafloor that have never been reached by cameras or sampling equipment. The environment is large enough, and the coverage limited enough, that such forms could exist without having entered the record. What has already been documented, however, provides a foundation that continues to deepen with each observation, revealing patterns that become more intricate as attention moves closer to the details of their structure and function. In this system, the work of continuation settles into a form that runs counter to what is commonly seen across most animal life. The burden does not rest where it is usually placed. Instead, it shifts, redistributing effort in a way that reshapes the entire rhythm of reproduction, placing the long responsibility of care onto a body already built for carrying. The process begins simply enough. Females release their eggs into the surrounding water, where fertilization occurs outside the body. There is no hidden chamber, no enclosed space in which development begins. The early stages unfold in direct contact with the environment, exposed yet immediately gathered into a more controlled arrangement. Males move through this moment not as distant contributors, but as central participants, collecting the fertilized eggs and securing them onto specialized appendages that extend from their bodies. These appendages, known as ovidures, are narrow and deliberate in their design, shaped for a single purpose that defines their existence. They hold the eggs in clustered masses, positioning them along the length of the limb where they can be maintained over time. Once attached, the eggs remain there, carried continuously as the male continues to move, feed, and navigate the seafloor. The body that already distributes its internal systems into its limbs now extends that pattern outward, turning those same structures into a cradle for the next generation. In Antarctic waters, where low temperatures slow every biological process, this period of care extends far beyond what would be expected in warmer environments. Development proceeds at a measured pace, unfolding across months that can span an entire winter. During this time, the male does not withdraw or isolate. He continues his movements, maintaining the same patterns of feeding and interaction while supporting the growing embryos. The presence of the eggs becomes a constant, integrated into every action rather than interrupting it. In some cases, a single individual may carry multiple clusters of eggs originating from different females, holding them simultaneously along the same appendages. Each cluster develops alongside the others, forming a layered arrangement that transforms the animal into a moving nursery. The genetic origins of these embryos may differ, yet they share the same support, sustained by the same steady care as they progress through their early stages. The attachment itself is not passive. The male maintains the eggs through subtle, continuous actions, adjusting their position, allowing water to flow between them, preventing accumulation that might hinder development. This care remains ongoing, a quiet sequence of movements that ensures each embryo remains viable within the conditions surrounding it. The process does not draw attention, yet it persists with consistency over extended periods. When development reaches the point of emergence, the larvae that appear are small and delicate, known as proto-nymphan larvae. They begin with only 3 pairs of legs, their bodies compact and nearly transparent, difficult to distinguish without close observation. At this stage, independence is not immediate. In many Antarctic species, the larvae remain associated with the male, clinging to his body for a further period as they continue to develop. They move with him, carried through the same environment in which they will eventually grow. From this beginning, growth proceeds slowly. The cold water does not permit rapid change. Instead, development stretches across years, possibly many years, as each individual gradually approaches its full size. The time between emergence and reproduction becomes extended, each stage unfolding within a system that rewards persistence rather than speed. This entire sequence depends on a consistency that allows such long durations to remain viable. The months of carrying, the extended development, the delayed maturity, all rely on conditions that do not shift abruptly. The environment supports a strategy built on patience, where continuity across time becomes more valuable than rapid cycles. Within this steady progression, the scale of time begins to extend beyond individual lives, accumulating across generations in a way that reflects the deeper continuity of the system itself. There comes a point in the history of a place when change stops being temporary and becomes a condition, something that settles in and remains long enough to shape everything that follows. When the Southern Ocean closed in on itself and the currents began their endless circuit, the shift did not arrive as a single event, but as a gradual narrowing of possibility. The water cooled, the connections faded, and the organisms already present found themselves within a system that no longer resembled the one they had known. What followed was not a moment of decision, but a filtering. Lineages that could not function under the new conditions receded from the record, leaving behind those that could persist. Survival did not require innovation in every case, but it demanded compatibility with a colder, more stable environment that would not return to its previous state. The forms that remained continued, generation after generation, adjusting within the limits of what was now constant. Across approximately 34 million years, this continuity produced a degree of separation that is rarely seen on such a scale. Antarctic marine species began to diverge from their relatives elsewhere, not through sudden transformation, but through the slow accumulation of differences that were never interrupted by external exchange. The passage of time itself became a defining force, allowing small variations to extend outward into distinct identities. This divergence can be traced through the subtle record held within genetic material, where patterns of change accumulate at a steady pace. By comparing these patterns, it becomes possible to estimate when lineages separated using what is known as a molecular clock. In many Antarctic sea spider species, these analyses suggest that their closest relatives outside the region diverged between 10 and 30 million years ago. Such spans place their separation far back within geological time, well beyond the scale of recent adaptation. These are not variations within a shared species, nor slight adjustments to a common form. They represent independent lineages, shaped in isolation for durations that exceed many familiar geological landmarks. Their histories extend back further than mountain ranges that appear permanent when viewed from a human perspective, yet are themselves transient over such spans. The increase in size observed within these organisms is not a recent response to changing conditions. Evidence from the fossil record indicates that larger forms were already present in Antarctic waters several million years ago. The pattern of expansion has been established for a long time, continuing across generations that have experienced no interruption in the underlying conditions that support it. The body plan itself reaches even further back. The oldest confirmed sea spider fossil, dated to approximately 425 million years ago, reveals a form that is immediately recognizable. It is small, its proportions modest, but its structure is already in place, the same arrangement of limbs and central trunk that persists in present-day species. The difference lies not in the design, but in the environment in which that design operates. Those early forms existed in warmer seas, where oxygen availability was lower and the limits on size were more restrictive. The architecture remained constant, but the scale was held within narrow bounds. When similar structures came to exist within colder, oxygen-rich waters, the constraints shifted, and the same basic form was able to extend further outward. The continuity of this design across such vast spans of time suggests a stability that reaches beyond any single environment. The organism itself does not require fundamental change to persist. Instead, it adjusts its scale and function within the conditions provided, maintaining its core structure while responding to the properties of the surrounding water. Over 425 million years, the same general body plan has continued, passing from one generation to the next without interruption. Over 34 million years, within the isolation of Antarctic waters, That plan has expressed itself in ways that differ markedly from its earlier forms. Together, these two spans of time converge in the present moment, where the lineage continues to move slowly across the seafloor, unchanged in its fundamentals, yet shaped by the long continuity of its environment. To understand how this influence extends beyond visible form, it becomes necessary to look more closely at the internal processes that allow these organisms to function under such conditions, where the effects of the environment reach into the smallest scales of their biology. In the quiet circulation of these waters, there exists a lineage of fish whose internal workings seem almost incomplete when measured against what is considered necessary elsewhere. Their bodies move through the same environment as countless others, yet the substance that flows within them lacks a feature so fundamental that its absence would normally bring life to an immediate halt. These are the Antarctic icefish, members of a family known as Channicthidae, a group composed of 16 recognized species. Within all known vertebrates, they stand alone in a remarkable way. They possess no functional red blood cells, and the molecule that defines oxygen transport in nearly every other vertebrate, hemoglobin, is entirely absent. Where most animals rely on the deep coloration of blood to carry oxygen efficiently through the body, these fish circulate a fluid that is almost transparent, pale, and thin, moving without the density or pigment that would be expected. In other contexts, such a condition would be incompatible with survival. Hemoglobin binds oxygen, allowing it to be transported in sufficient quantities to support active metabolism. Without it, the capacity of blood to carry oxygen drops dramatically. In warmer, less oxygen-rich waters, this limitation would prove fatal within a short span. The tissues would not receive enough supply to sustain even basic functions, and the organism would fail. Here, the surrounding water alters that equation. The concentration of dissolved oxygen is high enough that it can be absorbed directly into the plasma, the liquid component of blood, without the need for a specialized carrier. The oxygen enters, remains dissolved, and is transported through the body in this simplified form. The process is less efficient in absolute terms, but the environment compensates for that inefficiency by providing an abundance that keeps the system viable. To support this arrangement, the structure of the body adapts in ways that remain consistent with its internal logic. The heart becomes proportionally larger, increasing the volume of fluid moved with each contraction. The blood itself remains low in viscosity, flowing more easily through vessels and reducing resistance as it circulates. Together, these adjustments form a system that differs fundamentally from the standard vertebrate design, yet functions effectively within the specific conditions that allow it. This configuration does not represent a partial version of a more complete system. It is a distinct solution, shaped by a setting where the usual constraints have been removed. The absence of hemoglobin is not compensated by a direct replacement. Instead, the entire approach to oxygen transport is restructured, relying on the properties of the surrounding water rather than internal specialization. When placed alongside the long-limbed organisms already considered, a pattern begins to emerge that extends beyond individual groups. These fish and those distant arthropods share no recent ancestry. Their evolutionary paths diverged more than 500 million years ago, developing along entirely separate lines. Yet within this isolated environment, both have arrived at a similar functional outcome. Each has reduced or abandoned conventional respiratory structures, instead drawing directly from the oxygen available in the water itself. Such convergence does not arise from coincidence. It reflects a common influence acting across unrelated lineages, guiding different forms toward comparable solutions. The high concentration of dissolved oxygen becomes a central factor, shaping the possibilities available to organisms regardless of their origin. It is not simply a condition that supports life, but one that actively directs its form and function. In this way, the environment reaches inward, influencing systems at a level that is not immediately visible. The adjustments extend beyond size or structure, touching the fundamental processes that sustain each organism. What appears unusual when viewed in isolation becomes consistent when considered within the broader context of the surrounding water. Yet even this does not represent the deepest level at which these conditions exert their influence. Beneath these visible adaptations lies another layer, one that operates within the chemistry of living systems themselves, shaping how life functions at temperatures where ordinary processes would slow or fail. Deep within each cell, long before any structure takes form or any movement becomes visible, there is a constant exchange of reactions unfolding in sequences too small to observe directly. These reactions depend on molecules that act as guides, shaping and accelerating each step so that life can proceed at a pace that sustains itself. These molecules, known as enzymes, are not static tools but flexible structures, shifting their shape to bring other molecules together, enabling transformations that would otherwise occur too slowly to support life. Temperature influences this process at its most fundamental level. As the surrounding environment cools, molecular motion slows, and the interactions that drive these reactions become less frequent. In warmer conditions, enzymes can change shape quickly, facilitating reactions with ease. In colder settings, that flexibility diminishes, and the same processes begin to stall. For organisms accustomed to moderate or warm waters, a sudden transition to temperatures near -1.8 degrees Celsius would bring their internal chemistry towards stillness. Reactions would proceed too slowly, and the balance required for survival would begin to collapse. Over long spans of time, the organisms that remained within Antarctic waters adjusted not by altering their overall form, but by refining these molecular systems. Through gradual changes accumulated across generations, their enzymes became suited to function within the cold, maintaining activity at temperatures that would inhibit similar molecules elsewhere. The alterations are subtle, involving shifts in the folding patterns of the proteins that make up each enzyme. These shifts increase flexibility at low temperatures, allowing the molecules to continue changing shape and facilitating reactions even when the surrounding water is near freezing. This adaptation does not involve a complete redesign, but rather a precise adjustment, tuning the structure of each enzyme to operate within a narrow range of conditions. The same principle applies across different organisms, from those moving slowly along the seafloor to those swimming within the water column. Each has developed molecular systems that remain active where others would falter, sustaining the processes that support growth, movement, and reproduction. However, this tuning carries a limitation that is as important as its benefit. Enzymes that are optimized for cold conditions tend to lose stability when temperatures rise. The increased flexibility that allows them to function in the cold becomes a weakness in warmer environments, where the structures may begin to lose their shape, reducing their effectiveness or causing them to fail entirely. A small increase in temperature can disrupt the balance that has been maintained over long periods, altering reaction rates and affecting the stability of the entire system. This sensitivity is reflected in the behavior of organisms adapted to these conditions. When exposed to water that is only slightly warmer than their usual environment, they exhibit signs of stress, indicating that their internal processes are being pushed beyond the range for which they are suited. The change does not need to be large. Even a shift of 1 degree Celsius can influence the functioning of these systems, affecting the stability of enzymes and the reactions they control. The biochemistry of life in this region has been shaped by a temperature range that has remained consistent for millions of years. This consistency has allowed molecular systems to become highly specialized, operating efficiently within a narrow band of conditions. The result is a form of life that is not only adapted to the cold, but dependent on it, with processes that rely on the stability of the surrounding environment. These adaptations are not visible in the external form of the organisms. They do not alter the outline or the movement that can be observed from a distance. Instead, they operate at a level that remains hidden, influencing the behavior and survival of each organism through the functioning of its cells. The influence of the environment thus extends inward, shaping the chemistry that underlies every aspect of life in these waters. High above the seafloor, where the water meets the shifting boundary of ice, another layer of the system forms quietly, shaping everything beneath it long before its influence is visible. This upper surface does not remain inert. It becomes structured from within, threaded with narrow channels of brine that hold pockets of liquid within the solid matrix, creating spaces where life can persist even in conditions that appear uninhabitable. Within these channels, microscopic algae take hold, embedding themselves into the ice itself. They endure temperatures close to freezing and salinity levels that would disrupt most cellular systems, yet they continue to function, drawing in light that filters faintly through the surface and converting it into energy. Their growth remains modest during the darker months, sustained at a low level that maintains their presence within the ice. As the seasonal cycle progresses and light returns more consistently, the structure begins to change. The ice softens from below, releasing these algae into the surrounding water. What had been confined within narrow channels disperses outward, spreading through the upper layers of the ocean in a broad and coordinated pulse. This release does not occur in isolation. It unfolds across vast areas, seeding the water with organisms that rapidly multiply once freed into a more open environment. The result is a bloom of phytoplankton that extends across large sections of the Southern Ocean, forming a foundation upon which the entire food web depends. These microscopic organisms become the primary source of energy for larger forms, with krill feeding on them in dense aggregations, followed by fish and other animals that rely on the krill as a central resource. Each level builds upon the one below it, tracing a chain that begins with the algae embedded within the ice. Alongside this expansion of biomass, another process unfolds. Through photosynthesis, these organisms release oxygen into the surrounding water, increasing its concentration in the upper layers. This addition is not constant throughout the year, but occurs in pulses that correspond with the seasonal bloom. The oxygen produced at the surface does not remain there. It is carried downward through mixing processes driven by wind and the movement of water, extending its influence into deeper regions. This downward transport reaches depths of hundreds of meters, delivering oxygen to the layers where larger invertebrates reside. The timing of this input aligns with patterns observed in their growth, with periods of increased tissue formation occurring during and shortly after the peak of oxygen availability. The connection is not immediate, but it is consistent, linking events at the surface to changes occurring far below. The entire sequence depends on the presence and behavior of the ice. When its extent is reduced, the initial stage of the process is diminished. Fewer algae are present within the ice, leading to a smaller release when melting begins. This reduction carries through each subsequent step, resulting in a weaker bloom, a smaller increase in oxygen, and a reduced supply reaching deeper waters. Recent observations have recorded a notable change in this system. In 2023, the extent of Antarctic sea ice reached its lowest recorded level, falling by approximately 1 million square kilometers below the previous record. The reduction covered an area comparable in scale to Egypt, and unlike earlier fluctuations, it did not recover during the following winter period. The persistence of this change alters the baseline from which the seasonal cycle begins. With less ice present, the initial reservoir of algae is reduced. This leads to a cascade of effects that extend through the food web and into the deeper layers of the ocean. The influence of the ice becomes clearer when traced through these connections, revealing its role not as a passive boundary, but as an active component in the system that sustains life below. The reduction of this layer introduces changes that extend beyond a single process, affecting multiple aspects of the environment simultaneously. In a controlled space far from the open ocean, where variables could be adjusted with precision, a quiet test unfolded that would clarify something long suggested but not yet directly shown. The question was not whether large bodies could exist in these waters, but whether the presence of abundant oxygen was the cause of that scale, or simply a condition that accompanied it. To separate those possibilities required holding everything constant except the one factor under consideration. Researchers working within facilities connected to the British Antarctic Survey approached this by recreating the essential conditions of the Southern Ocean while carefully altering the concentration of dissolved oxygen. The temperature remained cold, matching the narrow range to which the animals were adapted. The organisms, including sea spiders and other invertebrates, were maintained in these environments as the oxygen levels were gradually reduced, not abruptly, but in measured steps that allowed the effects to emerge without interference from sudden change. What followed did not present itself as a subtle shift. Growth slowed in a way that was immediately apparent, with individuals failing to reach the sizes that would normally be expected under unchanged conditions. Development extended over longer periods, yet did not compensate for the reduced rate. Alongside this, mortality increased, even though the temperature remained stable. The system that had supported large forms began to falter when one component was altered, suggesting that the relationship between oxygen and size was not incidental, but central. This outcome provided direct experimental evidence that oxygen availability plays a determining role in shaping the scale of these organisms. It moved the understanding from observation to demonstration, showing that when the concentration of oxygen is reduced, the capacity for growth diminishes in a predictable way. The environment does not merely permit larger forms, it actively sustains them through a continuous supply that cannot be easily replaced. Alongside this confirmation, another pattern emerged, one that speaks to the history embedded within these organisms. When compared with species from more variable environments, those from Antarctic waters displayed a reduced ability to adjust to changes in oxygen levels. In regions where oxygen availability fluctuates, organisms often respond by slowing their activity, conserving energy, and enduring the period of scarcity until conditions improve. This flexibility allows them to survive temporary changes without lasting harm. In contrast, the Antarctic species showed a different response. When exposed to similar reductions in oxygen, they did not shift into a state of lowered activity. Instead, their systems began to fail more quickly, indicating that the capacity to cope with such changes had diminished over time. The consistency of their environment had removed the need for these adaptive responses, and without that pressure, the mechanisms that would support flexibility were no longer maintained. Further work conducted in subsequent years expanded on these findings, examining the relationship between oxygen levels and growth in greater detail. Studies led by researchers, including Amy Moran and H. Arthur Woods, revealed that the connection was not gradual in a simple sense, but marked by thresholds. Above certain levels of oxygen, growth proceeded within the expected range. Below those levels, the process did not merely slow, it ceased entirely. The organisms could not continue to develop under those conditions, regardless of how long they remained within them. These thresholds had been consistently exceeded in Antarctic waters for millions of years, allowing growth to proceed without interruption. The system had operated above its critical limits, supporting the expansion of body size across long spans of time. The experiment showed what happens when those limits are crossed in the opposite direction, when the supply falls below what is required to sustain the existing form. The implications of this extend beyond the controlled conditions in which they were observed. They suggest that the stability of oxygen levels is not only a background feature, but a defining factor that maintains the structure of the ecosystem. When that stability is altered, the effects propagate through the system in ways that are both immediate and far-reaching. Beyond this internal vulnerability, another change approaches from outside, moving through regions that were once separated by conditions that no longer hold. At the edge of the continental slope, where colder water once formed a boundary that few organisms could cross, a slow movement has begun to trace a new path inward. It does not announce itself in sudden numbers or abrupt change, but through the steady appearance of forms that were once held back by conditions that no longer remain as fixed as they once were. Among these arrivals are king crabs, animals whose feeding strategy reshapes the environments they enter. Their limbs are built not only for movement but for force, capable of grasping and breaking structures that resist softer forms of predation. They do not specialize narrowly. Their diet extends across a wide range of organisms, from those with hard external coverings to those composed of softer tissue. Sea urchins, brittle stars, mollusks, and other invertebrates fall within their reach, each vulnerable in different ways to the same set of tools. For long periods of time, the continental shelf remained beyond their range. The water was too cold for their early life stages to survive, creating a barrier that was not visible, yet consistently enforced. The threshold for their larvae lay above the temperatures found across much of the shelf, and so the populations remained confined to deeper or more temperate regions where those conditions could be met. This absence shaped the communities that developed in their place, allowing certain structures and behaviors to persist without the pressure that such predators would normally impose. As temperatures have shifted, even by small margins, that boundary has begun to move. Water that was once below the tolerance of these organisms now approaches levels that allow their larvae to survive. This change does not require a dramatic increase. A difference of a few degrees can alter the viability of early development, opening pathways that had remained closed for millions of years. Observations have begun to document this progression. Surveys using underwater cameras have recorded the presence of species such as Paralamus bursteini at depths shallower than approximately 850 meters along parts of the West Antarctic Peninsula shelf. These depths would have been inaccessible to such species in earlier periods, when the temperature profile of the water prevented their establishment. The change reflects not a shift in the animals themselves, but in the conditions that now allow them to occupy new areas. The communities into which they move have no recent history of encountering such forms of predation. Over approximately 34 million years, the absence of shell-crushing predators has influenced the development of these ecosystems. Many of the organisms that inhabit the shelf have not evolved the structural defenses that are common elsewhere. Sea urchins, for example, often possess thinner, more fragile tests, reflecting an environment in which the pressure to resist crushing has been minimal. In other oceans, where such predators are present, these structures tend to be thicker and more resistant, shaped by ongoing interactions that select for durability. The introduction of king crabs into this setting represents more than the addition of a new species. It brings with it an entire mode of interaction that has not been part of the system for a very long time. The effect extends beyond individual encounters, influencing the broader structure of the community as organisms that were once secure become accessible. The organisms that form the foundation of the seafloor communities, including the colonial structures that support many other forms of life, lie directly within the range of these predators. As these interactions begin to occur, the balance that has persisted over long periods becomes subject to new forces. The conditions that once maintained separation have shifted, allowing processes that were previously excluded to begin operating within this environment. The system that developed under one set of constraints now encounters another, altering the relationships that define it. These changes do not occur in isolation, but alongside other shifts that are unfolding at the same time, each influencing the system in different ways. Across the surface of the Southern Ocean, change does not arrive as a single disturbance, but as a gradual accumulation of shifts that settle into the system all at once. The water absorbs energy continuously, drawing in heat from the broader atmosphere and storing it beneath a surface that appears unchanged from a distance. Over time, this accumulation becomes measurable, not as a sudden spike, but as a steady increase that alters the baseline upon which everything else depends. Since the onset of large-scale industrial activity, the Southern Ocean has taken in a substantial portion of the excess heat generated across the planet, accounting for roughly 75% of that additional energy. This absorption exceeds that of any other ocean basin, placing the region at the center of a process that unfolds slowly but with persistent effect. The heat does not remain confined to the surface. It mixes downward, influencing layers that extend into the depths where long-established communities reside. In certain areas, sea surface temperatures have risen by approximately half a degree to 1 degree Celsius since the mid-20th century. On a human scale, this difference appears slight, a change that might pass unnoticed without instruments to record it. Within this system, however, such a shift carries greater significance. The organisms inhabiting these waters are adapted to a range that has remained within narrow limits for millions of years, with variation rarely exceeding 1 to 2 degrees Celsius. Their internal processes, from the behavior of enzymes to the balance of oxygen supply, are calibrated to operate within this confined interval. As temperature increases, the capacity of seawater to hold dissolved oxygen declines. For each degree Celsius of warming, the concentration of oxygen that the water can retain decreases by approximately 2%. This change does not occur in isolation. It accompanies the warming itself, meaning that the two factors arrive together, reinforcing one another. The environment becomes simultaneously warmer and less capable of supplying the oxygen upon which these organisms depend. The consequences extend through the system in ways that are both direct and cumulative. Models that examine the response of Antarctic invertebrates under scenarios of continued warming suggest that species specialized for cold conditions may experience significant reductions in their range by the end of the current century. These projections do not rely on a single variable, but consider the combined effects of temperature, oxygen availability, and ecological interactions, each contributing to a narrowing of the conditions within which these organisms can persist. Observations from recent years provide a glimpse of how these changes are already unfolding. In 2023, the extent of Antarctic sea ice reached a level that had not been recorded before, falling approximately 1 million square kilometers below the previous minimum. This reduction covered an area comparable in size to Egypt and persisted through the winter season that would normally restore the ice to its prior extent. The anomaly stood apart from earlier fluctuations not only in magnitude but in its persistence, remaining in place beyond the period during which recovery would be expected. The reduction in ice influences the system at multiple levels, altering the initial stages of production at the surface and the subsequent flow of energy and oxygen to deeper layers. At the same time, the movement of species from other regions introduces new interactions that further reshape the community. These processes do not occur in sequence, each waiting for the previous one to conclude. They overlap, acting together within the same period, compressing the system from several directions at once. The organisms within this environment have developed under conditions that favored specialization rather than flexibility. Their adaptations are precise, suited to a stable range that persisted over long spans of time. As that range begins to shift, the capacity to adjust becomes limited, reflecting the absence of pressures that would have maintained broader tolerance. Within this convergence of changes, the significance of the system extends beyond its immediate context, connecting to broader questions about the possibilities of life under conditions that differ from those found elsewhere on the planet. Far beyond the horizon of this ocean, where the sky gives way to distances that cannot be crossed by any current, there are other worlds whose surfaces conceal environments that bear a quiet resemblance to what lies beneath Antarctic ice. They are not places of open water and light, but of hidden oceans, sealed beneath layers of frozen crust, where conditions unfold in darkness and pressure over spans of time that extend far beyond human history. One of these worlds orbits Jupiter, a moon known as Europa. Beneath its outer shell of ice, there is strong evidence for a global ocean of liquid water, kept from freezing by the continuous flexing of the moon under the gravitational influence of the planet it circles. This process, known as tidal heating, generates enough internal energy to maintain a body of water beneath the surface, even as the exterior remains locked in ice. The ocean below is cold, shielded from sunlight, and isolated from any direct exchange with the surface for immense stretches of time. Another example lies in orbit around Saturn, where the moon Enceladus has revealed its internal activity through plumes of water vapor and organic compounds venting into space. These emissions suggest the presence of liquid water beneath its surface as well, interacting with rocky material in a way that produces chemical conditions similar to those found in certain deep marine environments on Earth. The combination of water, energy, and chemical complexity creates a setting that invites comparison with ecosystems that exist far from light. When scientists consider whether such environments might support life, they turn to analogs that can be studied directly. The Antarctic continental shelf provides one of the closest examples available, offering a system that shares key characteristics with these distant oceans. It is cold, covered by ice, removed from direct sunlight, and sustained by processes that do not rely on surface conditions. Within this setting, life persists in forms that are both complex and active, demonstrating that such environments can support more than simple organisms. The presence of large, mobile animals within this system carries particular significance. These organisms function without access to light, relying on energy pathways that originate elsewhere, and they obtain oxygen directly from the surrounding water rather than through specialized respiratory structures. Their existence shows that the absence of sunlight does not preclude the development of intricate biological systems, and that alternative pathways can sustain activity even under conditions that differ greatly from those found in more familiar environments. This does not imply that identical organisms exist beyond Earth. The forms that have developed here are shaped by the specific conditions of this planet, and their exact counterparts are not expected elsewhere. What their existence demonstrates is more general. It shows that certain combinations of cold, darkness, pressure, and chemical availability can support life at scales and levels of complexity that were once considered unlikely under such constraints. This understanding informs how missions are designed to explore these distant oceans. Instruments are developed with the expectation that if life exists, it may not resemble the forms found in sunlit environments, but may instead operate through processes similar to those observed in Antarctic waters. The questions guiding these missions are shaped by what has been learned here, where conditions once thought to be limiting have proven capable of supporting diverse systems. The exploration of these distant environments is guided not only by curiosity, but by evidence drawn from places that can be studied directly. The organisms inhabiting Antarctic waters provide a reference point, demonstrating that life can persist and adapt under conditions that extend beyond those typically associated with habitability. In this way, the system beneath the ice becomes part of a larger context, linking the study of Earth to the search for life elsewhere, and extending the relevance of what is observed here beyond the boundaries of this planet. Long before the outlines of forests rose on land, before roots pressed into soil or branches shaped the air, a different continuity had already settled into the oceans, moving slowly across the seafloor with a form that would endure across nearly the entire span of complex life. In those early seas, where continents were arranged in unfamiliar patterns and the surface above held no trees, the essential structure of the sea spider had already taken shape. The oldest confirmed fossil of this lineage comes from a deposit in what is now England, preserved within the Silurian Herefordshire Lagerstätte, a site known for capturing delicate marine organisms in remarkable detail. That fossil dates back approximately 425 million years, placing it in a world where terrestrial ecosystems had yet to assemble into anything resembling a forest. For tens of millions of years after that moment, land would remain largely bare of tall vegetation, while in the oceans this particular design continued without interruption. The structure itself is striking in its simplicity. A narrow, central body, so reduced that it cannot contain the full arrangement of organs found in most animals, extends outward into limbs that carry functions typically housed within a torso. A forward projection forms a rigid feeding tube, capable of extracting nutrients from other organisms, while specialized appendages are devoted entirely to holding and tending developing offspring. This arrangement was already present in those ancient seas, and it has persisted with only minor variation ever since. Across the immense stretches of time that followed, the planet underwent repeated transformations. The end of the Ordovician period brought a collapse in marine diversity that removed a vast portion of existing life. Later, during the Devonian, further losses reshaped ocean ecosystems again. The most severe event arrived at the close of the Permian period, when the majority of marine species vanished in what remains the most extensive extinction known. Subsequent crises, at the end of the Triassic and the end of the Cretaceous, continued this pattern, each one altering the composition of life in profound ways. Through each of these events, the sea spider lineage persisted. The body plan did not undergo a fundamental redesign. It did not shift toward complexity in the way many other groups did, nor did it abandon its unusual arrangement of organs and appendages. Instead, it remained consistent, moving through different oceans, adapting in distribution and scale, but retaining the same essential architecture. Fossil evidence from later periods reinforces this continuity. Specimens from Jurassic deposits in what is now France preserve soft tissue outlines that reveal animals almost indistinguishable from those found in Antarctic waters today. The proportions, the placement of structures, the overall configuration all remain recognizable across a span of more than 100 million years between those fossils and the present. This persistence does not arise from dominance in the conventional sense. These organisms are not defined by speed, nor by aggressive behavior, nor by complex strategies that allow them to outcompete others in rapidly changing environments. Their survival is tied instead to the kinds of places they occupy. They exist in regions that are often cold, removed from intense competition, and distant from the most disruptive forces that periodically reshape ecosystems elsewhere. Over time, the Antarctic environment became the most complete expression of those conditions. It provided isolation, stability, and a set of physical properties that aligned closely with the requirements of this ancient design. In that setting, the lineage continued without the pressures that drove more dramatic changes in other parts of the world, maintaining a continuity that stretches back to an era before terrestrial life had fully emerged. This long persistence places the present moment within a broader frame, where the conditions that once offered refuge are no longer fixed in the way they were for millions of years. And the stability that sustained such continuity begins to shift in ways that reach into the foundations of that history. Seen up close, the feeding structure extends forward with a kind of deliberate simplicity, a rigid projection held steady as the animal adjusts the rest of its body around it, as though the act of eating begins not with movement of the tool itself, but with the slow alignment of everything behind it. The form is unmistakable once noticed, a tube that appears almost geometric in its outline, tapering toward a tip shaped by 3 distinct plates that meet in a precise arrangement. This structure, known as the proboscis, serves as the sole interface between the animal and its food. It does not flex or curl, and it cannot reach outward in the way many feeding appendages can. Instead, it demands accuracy. The sea spider must approach its target with care, placing its legs in measured steps, adjusting its orientation until the tip of the proboscis meets the surface it intends to penetrate. Movement across the seafloor proceeds at a pace that allows for this level of control, often only a few centimeters each second, with pauses that stretch into stillness as alignment is refined. The prey in this environment does not flee. Colonies of hydroids and bryozoans rise from the substrate in intricate branching forms, while soft corals spread in layered structures that remain anchored in place. Each individual unit within these colonies is small, but together they form expansive fields of living tissue. The sea spider moves across these surfaces as though tracing a landscape, positioning itself over one polyp at a time. When the proboscis meets the surface, the 3-part jaw at its tip opens just enough to allow entry, piercing into the soft outer layer. From there, the feeding process becomes one of extraction. Internal fluids and tissues are drawn upward through the tube, leaving behind an emptied structure that remains fixed in place. The animal then withdraws, shifts slightly, and repeats the action on a neighboring unit. Over the course of a single feeding session, this sequence continues again and again, sometimes for hours, as the sea spider works its way methodically across the colony. The strategy itself is not unique in principle. Fluid feeding appears elsewhere in the animal world, most notably among terrestrial spiders, which similarly rely on external digestion and suction to obtain nutrients. Yet the resemblance is a convergence rather than a shared origin. These lineages are separated by vast evolutionary distances, and the similarity in feeding method arises from facing comparable constraints rather than inheriting a common design. What sets the sea spider apart is the persistence of this exact structure across immense spans of time. Fossil evidence indicates that the proboscis, in both form and function, has remained effectively unchanged since its earliest appearance hundreds of millions of years ago. While many other anatomical features in different groups have undergone repeated modification, refinement, or complete reinvention, this particular tool has endured with only subtle variation. There are other examples of long-standing biological designs. The compound eyes of horseshoe crabs continue to operate on principles established deep in the past, and the chambered shells of certain cephalopods reflect architectures that have persisted through multiple geological eras. Yet even among these, the continuity seen in the sea spider's feeding apparatus stands out for its directness. The same structural arrangement that existed in ancient oceans is still being used in the same essential way by animals living today. Each movement across the seafloor, each careful alignment, each insertion and withdrawal of the proboscis reflects a pattern that has repeated across countless generations. The setting has shifted, continents have moved, climates have altered, and entire ecosystems have risen and fallen. Yet this interaction between predator and colony has remained consistent in its form. In the cold waters beneath Antarctic ice, where temperatures hover just above the freezing point of seawater, the process continues without interruption, the animal advancing slowly, the proboscis held steady, the sequence unfolding as it has for an expanse of time that stretches far beyond ordinary human experience, carrying with it a continuity that is difficult to grasp in a single moment of observation. It becomes easier to speak about spans of time than to sense them, to arrange them into numbers and comparisons that sit cleanly on the page while remaining distant from any lived experience. A duration measured in hundreds of millions of years resists the ordinary scales through which human memory and imagination operate, slipping beyond the reach of familiar reference points and settling into abstraction. The lineage in question extends back roughly 425 million years, a continuity that began long before many of the forms now taken for granted had appeared. Written language, cities, and the entire arc of recorded history occupy only a thin edge of that expanse, a brief interval of roughly 5,000 years. Set against the longer duration, that entire record repeats tens of thousands of times within it, each cycle of rise and change compressed into a fraction of the whole. Looking further back, the sequence of major biological milestones begins to align differently. The earliest fishes with jaws emerged after this lineage had already taken hold in the oceans. The first insects to leave the ground and sustain flight appeared while it continued unchanged beneath the surface. The arrival of dinosaurs and their long presence across the planet unfolded within a world where this form had already persisted for vast stretches of time. Even the disappearance of those same dinosaurs, an event often treated as a boundary between eras, passed without interrupting that continuity. The difficulty lies not only in the scale, but in the uninterrupted nature of the sequence. At no point across that entire duration did the chain of reproduction break. Each generation produced the next, passing forward the same underlying design, adjusting in detail but never collapsing entirely. Through environmental upheavals that removed large portions of marine life, Through shifts in climate that restructured habitats on a global scale, the lineage continued, carried forward by countless individual events of survival and reproduction that linked one moment to the next. There is no single point along that chain that can be isolated as the origin of the present. Every individual alive now is connected through an unbroken succession of ancestors that stretches back across those hundreds of millions of years. The continuity is not symbolic or approximate, it is literal, a sequence of living transitions that has never ceased. Among the events that tested life most severely, the extinction at the end of the Permian period stands as the most extensive, removing the majority of marine species and reshaping ocean ecosystems almost entirely. Later intervals brought additional losses and reorganizations, each one altering the distribution of life and the relationships between species. Yet even within those periods of collapse, this particular lineage persisted, continuing through conditions that proved unmanageable for many others. The environments that supported that persistence were often those that remained somewhat removed from the most intense disruptions, regions where stability, even if relative, allowed continuity to hold. Over time, the Antarctic system became one of the most consistent of these settings, offering conditions that aligned with the requirements of organisms adapted to cold, stable waters. In the present moment, that continuity encounters a different kind of pressure. Rather than abrupt events that unfold over shorter intervals, the change arrives gradually, expressed through shifts in temperature and the properties of the surrounding environment. It does not resemble the rapid disturbances associated with earlier extinctions, yet it reaches into the same biological systems that have remained stable for extended periods. The scale of time involved makes the current transition difficult to place within an intuitive frame. The lineage has persisted through events that removed vast portions of life, yet it now faces conditions that alter the very environment that sustained it over tens of millions of years in its most recent setting. What comes into view is not only the potential loss of individual organisms, but the disruption of a continuity that has extended across nearly 500 million years, and the implications that carries for a system that assembled gradually over millions of years under conditions that no longer remain as they were. Across the seafloor, influence is not always visible in motion or scale, but in the quiet adjustments that ripple outward from a single presence, shaping patterns that extend far beyond the reach of any individual body. The sea spider, moving slowly among branching colonies, occupies a position that threads through multiple layers of this environment, its role defined less by dominance than by connection. As it feeds, it regulates the growth of the organisms it depends on. Hydroids and bryozoans form dense assemblages, each colony composed of many small units working together as a single structure. Left entirely unchecked, these colonies expand outward, covering available surfaces and competing with other species for space and resources. The sea spider's methodical extraction of tissue from one polyp after another introduces a limit, not by removing entire colonies at once, but by thinning them, shaping their boundaries, and preventing any single form from overwhelming the rest. This influence is not isolated. The same animal that grazes across these colonies is itself part of a wider exchange, moving within a system where it can also be taken as food. Certain fish species, along with some sea stars and cephalopods known from these waters, incorporate sea spiders into their own patterns of feeding. In this way, energy moves through the system, passing from one organism to another, each link depending on the presence of those before it. Even at the level of the individual body, the sea spider carries additional layers of interaction. Its external surface, the cuticle that forms the boundary between organism and environment, provides space for smaller forms of life to attach and persist. Microorganisms, early life stages of invertebrates, and other minute inhabitants occupy this surface, turning the animal itself into a moving substrate. The distinction between organism and habitat begins to blur as the sea spider becomes both a participant in the system and a place within it. These connections extend further still. The colonies that the sea spider feeds upon are not passive accumulations of tissue. They are active predators in their own right, capturing tiny drifting organisms from the water column. Those smaller organisms, in turn, depend on the productivity of microscopic plants that rise and fall with seasonal cycles. At each step, the flow continues, linking the movements of large animals on the seafloor to processes that begin far above them. In this arrangement, the sea spider occupies a position within a loop that folds back on itself. The oxygen that supports its size and physiology is partly generated by the same chain of biological activity that it helps regulate through feeding. Its presence influences the very system that sustains it, not in isolation, but as one element within a continuous exchange. Such patterns do not arise quickly. They are the result of extended periods of stability, where interactions can refine over time, settling into configurations that balance growth, consumption, and coexistence. The environment that allowed this system to assemble did so gradually, over millions of years, with each relationship adjusting in response to the others. Because of that history, the system does not exist as a collection of independent parts that can be removed and replaced without consequence. The connections are specific, shaped by the conditions under which they formed. There are no parallel communities elsewhere that mirror this exact arrangement, no external source from which identical relationships could be restored if they were lost. What remains is a network built through continuity, where each organism contributes to the structure that supports the others, and where the presence of one species carries implications that extend far beyond its immediate actions, forming a pattern that reflects the long processes that brought it into being and the delicate balance that maintains it. Seen from a distance that allows the full form to come into view, the span of each limb begins to read less as a feature of an individual animal and more as a record, a physical trace of processes that have unfolded across stretches of time, far beyond immediate perception. The size is not simply a matter of scale. It is an accumulation, a visible outcome shaped by conditions that have aligned and remained in place long enough to leave their imprint on living structure. Each extension of the body carries within it the history of repeated success. Feeding that could be sustained without interruption, reproduction that continued across generations, survival through intervals that removed other lineages, and the gradual tuning of internal processes to match an environment that remained remarkably consistent. These factors do not appear separately. They overlap, reinforcing one another, allowing growth to proceed where in other settings it would be limited. In most parts of the ocean, some of these conditions exist while others do not. Oxygen may be sufficient, but temperature fluctuates. Or food may be abundant, but competition and predation impose constraints that limit expansion. The balance shifts, and with it, the potential for size. Growth is checked, redirected, or held within narrower bounds. In the Antarctic system, those limitations aligned differently. Oxygen remained available at levels that supported diffusion across larger surfaces. Temperature stayed within a narrow range for extended periods. Food sources persisted in forms that could be accessed steadily, and the absence of certain types of predators reduced the risks associated with becoming larger. Time allowed these advantages to compound, generation after generation, without interruption. The resulting form reflects that continuity. It is not an anomaly that emerged suddenly, nor an exception that stands apart from expectation. It is consistent with what underlying biological principles would suggest when constraints are removed and favorable conditions persist. The extended limbs, the distribution of internal systems, the capacity to sustain a larger body, all follow from the interaction between organism and environment carried forward over long durations. What stands out is not the existence of such a form, but the rarity of the setting that allowed it to emerge. There is only one region on the planet where this exact combination of factors has remained in place for such an extended period. The conditions are not easily replicated elsewhere, and the outcome reflects that uniqueness. The perception of the sea spider as something unusual arises from comparison, from placing it alongside relatives shaped by different constraints. Within its own context, however, it represents a point along a continuum defined by the environment it inhabits. It is an expression of what becomes possible when the limits that typically govern growth are eased in a consistent and sustained way. In that sense, the size carries meaning beyond the individual organism. It points toward the conditions that produced it, offering a tangible representation of how physical and biological factors interact over extended periods. The form becomes a summary, not only of evolutionary history, but of the environment that guided that history at every step. Stepping back further, the focus shifts from the organism to the broader implication of its existence. The presence of such a form indicates that there are places on the planet where the usual constraints do not apply in the same way, where the rules that shape life elsewhere are adjusted by local conditions into something distinct. These places are not separate from the rest of the world, but they operate under a different balance of factors. Understanding this does not resolve into a single conclusion. Instead, it reframes the observation, moving from the question of how such a form appears to the recognition of what its existence reveals about the range of possibilities contained within the same planet, and about how much of that range has only recently come into clearer view. Work in these waters begins long before a sample is ever collected, unfolding through layers of preparation that must account for isolation, temperature, and distance in ways few other fields require. The route south narrows into a chain of dependencies: aircraft, ships, weather windows, each one aligning only briefly, carrying people and equipment toward a place where even routine movement demands careful negotiation with the environment. At the edge of the Ross Sea, McMurdo Station settles into its seasonal rhythm, expanding during the austral summer to house a population that approaches 1,000, then contracting through the winter months to a fraction of that number. From here, access to the seafloor begins with the ice itself. Divers cut through a surface that can be meters thick, opening a passage into water that remains just below the freezing point of fresh water, held liquid by its salt content. Entry is controlled, deliberate, with safety lines, communication systems, and layers of insulation that separate the human body from conditions it cannot endure unaided. Below that opening, every action becomes slower, shaped by equipment that behaves differently in near-freezing conditions. Materials stiffen, regulators respond with subtle delays, and the margin for error narrows. Time underwater is limited, not only by air supply, but by the need to manage heat loss and maintain precise coordination. What might be routine in other settings becomes a sequence of controlled movements, each one planned in advance. Further along the Antarctic peninsula, Rothera Research Station supports another line of work that unfolds across years rather than individual dives. Here, long-term monitoring provides continuity, allowing patterns to emerge from repeated measurements taken under consistent conditions. The work carried out in such settings depends on persistence, returning to the same questions across multiple seasons, building datasets that reflect gradual change rather than isolated observation. Field efforts extend beyond fixed stations. Expeditions move into regions that are visited only occasionally, where research vessels navigate through shifting ice and open water that can produce some of the largest waves on the planet. Remotely operated vehicles descend into depths that remain largely unseen, transmitting images and data from environments that are otherwise inaccessible. Each deployment carries its own uncertainties, shaped by currents, visibility, and the limits of the equipment itself. The process of studying living organisms in these conditions adds another layer of complexity. Specimens collected must be handled in ways that preserve their physiological state, often requiring immediate transfer into chilled seawater systems that maintain the temperatures to which they are adapted. Transport, whether between field sites or back to laboratory facilities, demands careful control, ensuring that even slight deviations do not alter the processes under study. Researchers working on these organisms have developed methods that accommodate these constraints, designing experiments that can operate within the limits imposed by the environment. Some work directly on site, using facilities adapted to the cold, while others rely on transporting organisms under controlled conditions that replicate their natural habitat as closely as possible. In each case, the objective remains the same: to observe processes that unfold slowly, often requiring extended periods to reveal their structure. Across these efforts, a pattern emerges that mirrors the systems being studied. Continuity becomes central, with progress built through repeated engagement rather than singular breakthroughs. The work is sustained through return, through the willingness to operate within conditions that restrict speed and demand patience. What draws individuals into this environment is not easily reduced to a single explanation. It involves an alignment between the questions being asked and the place in which those questions can be addressed. A recognition that certain aspects of the natural world can only be understood by going directly to where they occur. The commitment extends across years, sometimes decades, forming a continuity of effort that parallels the long processes unfolding in the system itself, carrying observation forward in the same quiet persistence that defines the environment under study. For most of the span that shaped this system, change arrived slowly enough to be absorbed without breaking continuity, allowing the surrounding conditions to function as a steady backdrop against which life could refine itself. In ecology, this steady backdrop is often described as a baseline, a set of environmental conditions that defines what is normal for a community, not in a general sense, but in the precise context in which it evolved. On the Antarctic continental shelf, that baseline held with an unusual degree of consistency. Temperatures remained low, oxygen stayed abundant, and the physical isolation of the region limited the arrival of outside influences. Over tens of millions of years, these factors did not fluctuate widely, and the organisms that persisted there adjusted to that constancy. Their internal processes, from the way enzymes function to how oxygen is acquired and used, became aligned with conditions that changed very little across immense stretches of time. This alignment carries an implicit assumption. It is not a conscious expectation, but it is embedded within the structure of each organism. The chemical reactions that sustain life, the rates at which tissues grow, the interactions between species, all proceed as though the surrounding environment will continue to provide the same conditions that shaped them. The baseline becomes part of the organism itself, expressed through its physiology and behavior. Natural selection operates within this framework. It refines organisms in response to conditions that are present, not conditions that have yet to appear. It cannot anticipate future states, nor can it prepare lineages for scenarios that have not occurred within their history. Adaptation, in this sense, is always a response to what has been, rather than a preparation for what might be. In the Antarctic system, this means that many of the species present have no history of coping with warmer water, lower oxygen availability, or the arrival of certain types of predators. Their biological systems have not been shaped by those pressures, and therefore do not contain mechanisms to manage them. The enzymes that operate efficiently in cold conditions begin to lose stability as temperatures rise, and the processes that rely on high oxygen availability become constrained as that availability decreases. The rate at which current changes are occurring introduces an additional constraint. Evolutionary adjustment requires time measured in generations, and the number of generations that can occur within a given period depends on how quickly a species reproduces. In environments where conditions fluctuate, many species complete multiple generations within short spans, allowing selection to act repeatedly and potentially guide adaptation. On the Antarctic shelf, life often proceeds at a slower pace. Growth is gradual, development extends over longer intervals, and reproduction may occur less frequently. This reduces the number of opportunities for selection to operate within the time frame over which environmental conditions are shifting. The capacity for adjustment is therefore limited, not only by the absence of prior exposure to new conditions, but also by the pace at which new generations can arise. This pattern is not confined to a single group. It extends across many of the organisms that inhabit this environment, particularly those that have become highly specialized through long periods of stability. The very features that define their success under consistent conditions, precise biochemical tuning, reliance on specific temperature ranges, and integration into established ecological relationships, also restrict their ability to respond when those conditions begin to change. What emerges is a contrast between the time required to build such a system and the time over which it can be altered. The processes that assembled this community unfolded gradually, shaped by stability that allowed interactions to settle and persist. When that stability shifts, the effects propagate through the same connections that once supported continuity, altering relationships that took millions of years to establish, and doing so within a span that falls well within the scale of a human lifetime. Long before it became a place of study, this region functioned as a quiet boundary, a space set apart by moving water and distance, where continuity could persist without interruption. When the encircling current formed, and the last connection to surrounding continents slipped away, the separation did more than isolate. It established a kind of shelter, not by intention, but by the simple consequence of geography and motion. The water that now circles the continent does not rest. It moves in an unbroken loop, a persistent barrier that limits exchange, filtering what can enter and what can leave. In doing so, it created conditions where the wider disturbances shaping the rest of the planet arrived diminished, if they arrived at all. Events that reshaped entire ocean basins elsewhere, shifts in temperature, collapses in food webs, cascading losses of species, reached this margin with less force, sometimes reduced to faint traces of change. Within that boundary, stability settled in. Temperatures remained low and remarkably consistent, avoiding the fluctuations that repeatedly reorganized ecosystems in other regions. The absence of strong seasonal extremes, combined with the steady presence of oxygen in the water, allowed biological systems to align with a narrow range of conditions. Over immense stretches of time, this steadiness became the defining characteristic of the environment, shaping not only which organisms could survive, but how they functioned at every level. In this sense, the region operated as a refuge. Not a refuge in the way it is often imagined, as a place actively shielding life, but as a consequence of isolation and constancy that reduced exposure to external disruption. Species that might have struggled elsewhere found continuity here, and lineages that persisted carried forward traits that reflected a long history without abrupt change. That history is written into the organisms themselves. Their physiology, their growth patterns, and their interactions all bear the imprint of conditions that did not demand rapid adjustment. Flexibility, in many cases, was not favored because the environment did not require it. Instead, precision became the dominant theme, with biological systems tuned closely to a stable set of parameters that held across generations. There is a quiet weight to this outcome. The absence of frequent disruption allowed complexity to accumulate, but it also meant that the mechanisms needed to respond to sudden change were not reinforced. Adaptability, which in other environments can serve as a buffer against variability, remained limited where variability was rare. The organisms that emerged from this setting are, in a sense, records of success under stability, carrying forward designs that were never tested against rapid shifts. This makes the idea of refuge more intricate than it first appears. Protection over long durations can shape life in ways that depend on the continuation of that protection. When the conditions that provided stability begin to move outside their historical range, the same traits that once ensured persistence may no longer align with what is required. The separation that once limited disturbance does not prevent change within the system itself. Alterations in temperature, in oxygen levels, and in the broader dynamics of the surrounding ocean can still reach inward, reshaping the environment from within rather than through external intrusion. The boundary remains, but its character shifts as the properties of the water it contains begin to change. In this way, the refuge is not removed from the larger processes affecting the planet. It is connected through the medium that defines it, the moving water that both isolates and links it to distant forces. What was once a buffer can become a conduit, carrying gradual changes into a system that has not experienced them in its long history. The organisms that persist there embody a long period during which their assumptions about the world held true. Their continued presence reflects an unbroken sequence of conditions that allowed those assumptions to remain valid. As those conditions begin to shift, the continuity they represent encounters a different kind of test, one that unfolds not through sudden events, but through sustained change that moves steadily across the baseline that once defined their world. Across the length of this story, a quieter layer has been forming beneath the details, something less visible than anatomy or temperature, yet present in every part of it. It emerges not from any single species or process, but from the scale of time that surrounds them, a scale that does not align with how human lives are measured or understood. Human civilization, from the first settled communities to the present, spans roughly 10,000 years, a duration that feels expansive when held against memory and history. Entire cultures rise and fall within that window. Languages form and dissolve, landscapes are reshaped, and knowledge accumulates in ways that feel immense from within the human perspective. Yet, set against the lineage that produced the animals moving across the Antarctic seafloor, that entire span becomes a brief interval, a small segment within a much longer continuity. The lineage itself extends back roughly 425 million years, a stretch of time so large that repetition begins to lose meaning. If the entirety of human civilization were placed end to end across that duration, it would repeat tens of thousands of times, each cycle beginning and ending while the lineage continued uninterrupted. The comparison does not diminish human history, but it places it within a broader frame, one where persistence operates on scales that are difficult to hold in a single thought. Within that frame, the ecosystem that allowed these organisms to become what they are today assembled over roughly 34 million years. It was not constructed quickly, nor through sudden transformation, but through the gradual alignment of conditions that remained stable long enough for small advantages to accumulate. Each generation added a slight refinement. Each interaction adjusted the balance of the system until what exists now took shape through the steady passage of time rather than through abrupt change. This introduces a question that sits beneath the scientific narrative, one that does not resolve easily within familiar ways of thinking. Ethical systems, as they are commonly applied, tend to operate within spans that match human experience, focusing on individuals, communities, and perhaps a few generations into the future. They are effective within those bounds, but they are not naturally equipped to evaluate processes that unfold across tens of millions of years. The difficulty lies not in recognizing value, but in measuring it. When something has taken 34 million years to form, its significance is not captured by the same reasoning used for systems that can be rebuilt within a few decades or even centuries. The conditions that produced it, the isolation, the temperature stability, the continuous presence of oxygen in the water, and the absence of certain pressures, are not easily recreated once disrupted. They are the result of a particular configuration of the planet that unfolded over geological time. What is at stake, then, is not only the presence of individual species or even the structure of a single ecosystem. It is the continuity of a process that has been running for a duration far beyond human experience, a process that cannot simply be restarted if it is interrupted. The loss, if it occurs, would not resemble the kinds of change that can be reversed within historical time. It would represent the ending of a long sequence that cannot be reconstructed within any span that human civilization can meaningfully observe. There is a quiet tension in holding this understanding. The arithmetic of deep time is clear, yet it does not translate easily into instinct or feeling. It remains abstract, even as its implications are concrete. The organisms moving slowly across the seafloor carry within them the accumulated outcome of that time, not as a record that can be read directly, but as a living continuation of it. To consider what is owed to something that exists on this scale requires stepping beyond the boundaries of familiar thought, not to resolve the question completely, but to recognize that it exists at all. The presence of such systems on this planet suggests that time itself can act as a creative force, given stability and the absence of interruption, producing forms and relationships that cannot be rushed or replicated. In this sense, the story becomes less about any single species and more about the existence of a process that has been allowed to unfold without being broken. What follows from that recognition leads naturally toward a final reflection, one that gathers these threads without forcing them into a simple conclusion, and allows the scale of what has been described to remain present as the narrative approaches its quiet close. At the edge of this story, the word itself begins to shift, taking on a different weight than it carried at the beginning. It has followed quietly through each section, attached to an image of long limbs moving across a dim seafloor, but its meaning has remained unexamined until now, shaped more by instinct than by understanding. Historically, the word monster described something that stood outside expectation, something that seemed to break the rules governing how living things were meant to exist. It implied a deviation, an anomaly, a form that could not be easily explained by the patterns people recognized in the world around them. That definition carries forward into the present, even when the language has softened, leaving behind a subtle assumption that what appears unfamiliar must somehow be unnatural. Yet, the animal at the center of this narrative does not fit that description when viewed through the lens that has been gradually assembled. Its structure, its size, its method of breathing and feeding, all of it follows directly from the same principles that shape every organism on the planet. There is no break in the logic, no step where the process deviates from the rules. Instead, there is a continuity so consistent that the result appears strange only because the conditions that produced it are rarely encountered. The sense of strangeness comes from contrast. Most human experience is grounded in environments where temperature fluctuates, where oxygen availability varies, where predation imposes limits on size, and where ecological relationships are continually being reshaped. Within those contexts, certain boundaries appear stable, forming an internal model of what is typical. When an organism exists outside those boundaries, it can seem as though it has stepped beyond what is possible. In reality, it has simply followed the same rules further than expected. Remove constraints that normally restrict growth, allow stability to persist across immense spans of time, and the outcomes shift accordingly. The proportions change, the scale expands, and what once seemed like a boundary reveals itself as a local condition rather than a universal limit. This reframing places the animal not at the margins of biology, but at its center, as a clear expression of how environment and time interact to shape living systems. Every species reflects its surroundings, carrying within its form the imprint of pressures and opportunities that have acted across generations. The difference here is one of visibility. The outcome is large enough, distinct enough, to make the underlying process easier to see. Seen this way, the term monster begins to lose its relevance. It no longer describes a departure from order, but a misunderstanding of it. What appears unusual is not a violation of natural law, but an extension of it into a set of conditions that are rarely part of everyday experience. The unfamiliar becomes a reminder that the range of possible forms is wider than the narrow slice most often observed. There is also a subtle shift in perspective that follows from this recognition. The idea of normality, often treated as a fixed reference point, reveals itself as something shaped by familiarity. What is called normal is simply what is encountered most often, not what is fundamentally representative of life as a whole. In different conditions, different expressions emerge, each one equally consistent with the same underlying principles. The organism moving slowly across the seafloor does not exist to challenge those principles, but to demonstrate them in a context where their effects become more pronounced. It is, in a sense, a clearer version of the same process that produces all life, a form that reflects its environment with minimal distortion. As the narrative approaches its close, the image of the animal remains, but its meaning has shifted. It is no longer framed by distance or unfamiliarity, but by continuity, by the recognition that what seemed extraordinary is part of a broader pattern that extends across the planet. What remains to be considered lies not in redefining the animal, but in reflecting on what this wider view suggests about the world that produced it and the place within that world from which it has been observed. A beam of light moves slowly across a surface that has not seen sunlight in millions of years, tracing over structures that rise from the seafloor like delicate frameworks. each branch holding its place in water that rarely shifts beyond its narrow range. The motion is deliberate, controlled, a quiet sweep that reveals texture rather than searching for spectacle. Below the ice, the temperature rests just below the point at which seawater would freeze without salt, a condition that defines everything present. The diver's breath forms a steady rhythm contained within equipment designed to resist the cold, while the surrounding water remains still, carrying only faint currents shaped by forces far above. Visibility is limited, not by obstruction, but by the simple absence of light, leaving the beam as the only window into what lies ahead. The torch passes over a section of rock covered in fine branching forms, hydroid colonies extending outward in patterns that suggest growth measured over long intervals. Their surfaces hold small details that shift gently as the water moves, each part connected to the larger structure that supports it. The scene is quiet, composed, without urgency. Then something changes within that frame. A movement appears, subtle at first, not separate from the background but emerging from it. A rearrangement of lines that resolves into a form. 8 legs extend outward from a central body, each segment moving with measured precision, contacting the surface and lifting again in a sequence that is unhurried and continuous. The body itself is compact, no larger than a human fist, while the span of the legs reaches outward beyond it, creating a silhouette that occupies more space than its core would suggest. The proportions feel unfamiliar, not because they are irregular, but because they do not match the expectations carried into the scene. The animal moves across the surface with a pace that reflects the environment, neither fast nor slow, but consistent. The diver does not retreat. The movement of the light steadies, holding the form within its beam. There is a pause, not driven by hesitation, but by attention. The legs are counted, not as a reflex, but as an act of observation, a way of confirming what is being seen within a context that is still forming. This moment, simple in its structure, carries a response that extends beyond the immediate encounter. Stillness replaces motion, and observation replaces reaction. The impulse is not to interpret quickly, but to remain with what is present, allowing its details to settle into understanding without forcing them into familiar categories. The animal continues its movement, unaffected by the presence above it, following a path defined by its own processes. It carries no indication of the duration that produced it, no signal of the continuity that extends far beyond the moment. Its actions are local, immediate, part of a sequence that has repeated across generations without interruption. Yet within the diver's awareness, that continuity is present. The understanding of what has been observed is layered with context, not imposed on the animal, but held alongside it. The movement across the rock face is not only a behavior, but part of a longer pattern that connects the present moment to a history that stretches far beyond it. Attention, in this sense, becomes more than observation. It is a way of aligning perception with scale, allowing something that exists across vast spans of time to be recognized within a brief encounter. The act of watching, of counting, of remaining still becomes a method of bringing that scale into focus, not by expanding the moment, but by deepening it. The diver's response does not alter the animal, nor does it change the environment in which it moves. What it changes is the meaning of what is being seen, transforming it from an unfamiliar form into a clear expression of processes that have been unfolding continuously. The scene remains quiet, unchanged in its physical details, yet its significance expands through the act of sustained attention. In that space, between the movement of the animal and the awareness of the observer, something settles into place. The image holds, not as a fleeting encounter, but as a point where observation and understanding meet, preparing the ground for the final reflection that follows, where the question is no longer how something appears, but what it is when seen without distance or assumption. Set aside the scale, the history, the framing that has gathered around it, and let the animal remain on its own terms, occupying a single moment in a place that does not change quickly. What remains is not an abstraction, but a sequence of simple actions, each one repeated across a span of time far beyond any individual life. It moves. The motion is steady, measured against the surface. Beneath it, each leg placed with precision, lifted, and placed again. The pace falls within a narrow range, a few centimeters in the space of a second, consistent with the environment that surrounds it. There is no urgency in the movement, only continuation, a progression that carries it across ground it has never left and will not leave. It breathes. The exchange happens across the outer surface of its body, where dissolved oxygen passes into its tissues directly from the water. The temperature of that water rests just below the point where it would freeze without salt, a condition that holds more oxygen than warmer seas. There are no specialized organs guiding this process, only a surface that allows it to occur continuously as the animal moves. It feeds. The structure used for feeding extends forward, rigid, shaped to interact with the organisms fixed to the surface. The animal positions itself carefully, aligning its body with what lies ahead, then draws nutrients from colonies that do not move to avoid it. The method has remained consistent across immense stretches of time, a design that persists because it continues to function within the conditions that surround it. It carries forward its lineage. In the case of a male, this occurs visibly through clusters of eggs held on appendages that are also part of its locomotion. The same limbs that move the animal across the seafloor also support the next generation, maintaining contact as development proceeds slowly in the cold. The young remain attached through early stages, then release into the same environment, beginning a progression that will take years before reaching maturity. All of this unfolds in darkness. The light that reveals it is temporary, introduced from above, while the environment itself remains without direct illumination. Above, layers of ice extend across the surface, separating this space from the air, shaping the conditions below in ways that persist across seasons and years. The pressure, the temperature, the composition of the water all combine to define the limits within which the animal exists. There is no variation in behavior that suggests awareness of scale or history. The actions remain local, immediate, repeated as they have been repeated across countless generations. The continuity lies not in memory, but in process, in the unbroken sequence of movement, feeding, breathing, and reproduction that connects each individual to those that came before. The duration of that sequence extends back to a time before forests covered land, before the forms now familiar had appeared. Yet in the present moment, none of that duration is visible in the animal itself. It is carried implicitly, embedded in the structure and function that allow it to exist as it does. But it does not alter the way the animal moves across the surface beneath it. There is no performance in these actions. They are not exaggerated or displayed, but simply carried out, consistent with the conditions that make them possible. The surrounding environment does not demand speed or adaptation to rapid change, and so the behavior reflects that steadiness, proceeding without interruption. In this view, the animal is not defined by the context that has been built around it, but by what it is doing in this moment. A sequence of processes aligned with an environment that has allowed them to persist without alteration, continuing as they have for a span of time that exceeds ordinary comprehension. This is what life becomes when it is given stability, when the parameters within which it operates remain constant long enough for its processes to refine themselves without being reset. The result is not dramatic when seen up close, but it is precise, each part functioning within a system that has had time to settle into its current form. The animal continues its movement, the same slow progression across the surface, unchanged by observation, carrying forward a pattern that does not require acknowledgement to persist. Far from the reach of wind and sky, a vast plate of ice extends across the Southern Ocean, covering an area on the order of 500,000 square kilometers, a span comparable to an entire nation. Beneath that surface, the water holds steady just below freezing, a temperature that has remained within its narrow range across spans of time that stretch beyond ordinary comprehension. Light does not reach this space, and has not for as long as the ice above has persisted, leaving the seafloor in a continuous darkness shaped only by the faint glow of rare intrusions. Within that darkness, movement continues. Sea spiders traverse the seafloor at this moment, not as an idea or a reconstruction, but as a present reality unfolding beyond direct human sight. Their motion is unhurried, their pace aligned with the environment that surrounds them, each step part of a sequence that has repeated without interruption across generations that extend far beyond any recorded history. They move as they have always moved, placing each leg in contact with the surface, lifting and placing again, progressing across terrain that has changed little within the span of their lineage. They feed in the same manner, drawing sustenance from organisms fixed in place, interacting with the same kinds of structures that have defined their environment for millions of years. They breathe through their outer surface, extracting oxygen from water that holds it in abundance due to the cold that defines the region. Above them, the conditions that have allowed this continuity are beginning to shift. The ice that once maintained a stable boundary shows signs of retreat, its extent changing in ways that alter the system below. The temperature of the surrounding water rises gradually, affecting the balance that has held for so long. The movement of species from other regions introduces new interactions, and the concentration of oxygen, once consistently high, begins to adjust in response to these changes. The outcome of these shifts remains uncertain. The animals that inhabit this environment may persist, adapting in ways that are not yet fully understood. They may change in size, their forms adjusting as the conditions that supported their growth alter over time. They may withdraw from certain areas, their presence receding from parts of the seafloor that have long been within their range. It is also possible that some lineages will not continue within this environment, their long continuity coming to an end under pressures they have not previously encountered. What remains clear is the nature of what exists now. The organisms moving through this space represent a continuity that extends across hundreds of millions of years, a sequence of reproduction and survival that has not been broken. Their structure reflects a lineage that has persisted through multiple global changes, maintaining its form while the conditions around it shifted in ways that did not disrupt its fundamental processes. That continuity continues into the present. The chain has not been interrupted, and the processes that define these animals are still unfolding as they have for vast stretches of time. The movement across the seafloor, the feeding, the breathing, and the carrying forward of the next generation all proceed within the same framework that has sustained them. There is a sense of scale embedded in this, one that does not require amplification or emphasis to be recognized. The duration involved, the stability that allowed it, and the processes that continue within it form a combination that is rarely encountered. It does not demand a particular response, but it does invite attention, a recognition of what exists within a part of the world that remains largely unseen. The cold and the darkness beneath the ice hold forms that align precisely with the conditions that created them. They are not deviations from expectation, but expressions of it, shaped by the same principles that govern all life, extended into a context that is unfamiliar but consistent. They have been present within this space for a span of time that exceeds ordinary reference, and they remain there now, continuing in the same manner as before, part of a system that has persisted across the longest scales available to life on this planet.